Circular emergence, clinical consequence. Welcome back to parasitology. Hi, I'm Christopher. In this video, we move into the Cyclorrhaphan dipterans, the higher flies. The name itself teaches. Cyclo means circular, and rhaphe refers to a seam. In many of these flies, the adult emerges from the puparium through a circular seam or opening. Clinically, this group is enormous. It includes house and stable flies, tsetse flies, keds, flesh flies, blowflies, screwworm flies, and bot flies. Some are mainly nuisance flies, others bite, some transmit pathogens, and some have larvae that invade wounds, skin, nasal passages, or even the gastrointestinal tract. Your goal is to be able to identify the fly, understand its behavior, and predict the clinical consequence. Before we zoom in, let's place Cyclorrhaphans on the dipteran roadmap. Remember nematocerans, the long antenna flies include mosquitoes, black flies, biting midges, and sandflies. Tabanids, the horseflies and deer flies, sit in the non-Cyclorrhaphan brachyceran teaching bin. Today, we are inside Cyclorrhapha, the higher flies, nested within Brachycera. A taxonomy note. This roadmap is a teaching framework, not a complete phylogeny lesson. For veterinary reasoning, it works because it helps us organize vector biology, feeding behavior, larval habitats, myiasis risk, and clinical relevance. This chart compares the main dipteran groups. Nematocerans often have those long, multi-segmented antennae and will develop in aquatic or very moist habitats. Tabanids have short antennae, painful bites, and often have mechanical transmission patterns. For both nematocerans and non-Cyclorrhaphans, only the female typically feeds off blood. Cyclorrhaphans boast short antennae, which are often adorned with an arista. Their larval development is more varied and can occur in carrion, feces, decaying organic matter, wounds, tissues, nests, or even on the host. Their relationship with the host ranges from a temporary nuisance visitor to close host association to obligate parasites. Remember these patterns, but know that they are not an absolute rule. Nematoceran vectors are often biologic, tabanids are more commonly mechanical, and Cyclorrhaphans vary by family. Let's examine the face of a Cyclorrhaphan, and honestly, she is beautiful. Notice the dicoptic eyes. Dicoptic simply means that the compound eyes are separated. In many flies, separated eyes suggest that we are looking at a female, while males are often more holoptic, with the eyes closer together or even touching. That is a useful rule, but not an absolute. Now look at the arista, the bristle-like projection coming off of the antenna. The arista is a sensory structure. It can help the fly detect environmental cues such as air movement, vibration, sound, temperature, and even humidity. For us, it is also a wonderful identification clue. If you hear yourself thinking short antenna plus arista, you're starting to think like a fly diagnostician. Fun etymology: arista comes from the Latin word for on or beard of grain, meaning a stiff, bristle-like projection. Another important feature is the talinum and the talinal suture. During adult emergence, many Cyclorrhaphans use an inflatable structure called a talinum to help push open the puparium. The group name Cyclorrhapha refers to a circular opening or seam in the puparium through which the adult emerges. After emergence, the talinum retracts back into the head, leaving the talinal suture as a visible seam on the adult fly's face. If we're being poetic, the talinal suture is a little emergent scar, not a true belly button, but a visual reminder of how the adult fly escaped the puparium. Here's the framework for the rest of the video. Cyclorrhaphans create 4 major veterinary problems. First, nuisance and production loss. Even flies that are not major vectors can cause irritation, stress, reduced feeding, decreased milk production, and welfare concerns. Second, pathogen transmission. Some flies move agents mechanically on contaminated mouthparts, feet, or body surfaces. Others are true biologic vectors or intermediate hosts, where the agent develops as part of its life cycle. Third, myiasis. This is where larvae invade wounds, skin, body cavities, or tissues. And fourth, non-biting secretion vectors. Not every important fly feeds on blood. Some exploit tears or ocular secretions, which opens up the door to parasites such as Thalasia. We will start with the family Muscidae. The name Musca is wonderfully direct, as it simply means fly. This family includes familiar non-biting nuisance flies and several important blood-feeding muscids. The non-biting group includes the housefly, Musca domestica, the facefly, Musca autumnalis, and Phania species. These flies may feed on secretions, waste, or decaying organic material, and they can mechanically move pathogens onto feed, water, surfaces, and animals. A quick mnemonic for Phania canicularis, the lesser housefly. Now, I would not treat this as a strict etymology, but the name gives us a useful memory hook. Phania can remind us of a Roman feminine name, and historically, one Phania has been discussed as a model of a good Roman woman. Then, canicularis points us towards the dog days of summer. So picture it, a proper Roman woman annoyed by a small housefly during the hot dog days of summer. Silly? Yes. Sticky? Also yes. This image helps us remember Phania canicularis as the lesser housefly, a less famous but common fly around people, animals, and organic material. The biting muscids include the stable fly, Stomoxys calcitrans, and the hornfly, Hematobia irritans. The stable fly, Stomoxys calcitrans, has a wonderfully honest name. Stomoxys comes from the Greek roots meaning sharp mouth, which fits the painful, piercing mouth parts, and calcitrans meaning kicking, a perfect image for animals stomping and kicking at biting stable flies. The hornfly, Hematobia irritans, is just as direct. Hematobia points us towards blood feeding, and irritans means irritating, which is exactly what hornflies do when they cluster on cattle and repeatedly blood feed. And of course, if you are wondering whether there are many more muscid flies being left out of this image, you are absolutely correct. There are thousands of muscid species. This image is only a clinical entry point into the world of Cyclorrhaphans. Stomoxys calcitrans, or stable flies, feed on the host, often around the lower legs, and then leave rather than remaining on the animal. That matters clinically because the animal still experiences pain, stress, and production loss, but control cannot focus only on treating the host. You also have to think about the environment, especially moist, decaying organic material, manure, bedding, spilled feed, and other larval development sites. Hematobia irritans, the hornfly, is smaller, usually about 2.5 to 4 millimeters. It has shorter proboscis with about equal in length, and it remains on the cattle for much of its adult life. That host association changes the clinical picture because the animal experiences repeated irritation and feeding pressure. It also changes control strategy. Because hornflies stay on the host, management often focuses on treating the animal directly. Using tools such as insecticide ear tags, pour-ons, sprays, or forced use dust bags and back rubbers placed where cattle must pass through on their way to water, feed, or mineral. The maggot spherical table at the bottom is useful for recognition, but it is not the main thing to memorize today. For clinical pattern recognition, focus on stable fly versus hornfly behavior. Muscidae can feel messy because one family contains several different veterinary stories. Filth flies, such as Musca domestica and Phania, breed in filth and decaying matter. That's important because they can contaminate feed, water, and surfaces. Dung-associated and host-associated flies such as Hematoba irritans and Musca autumnalis, keep us thinking about cattle, horses, manure, secretions, and irritation. Sweat and secretion-associated flies, such as Hydrotia species, remind us that the head, neck, and eyes may also be important body sites. Then the biting muscids, especially Stomoxys and Hematobia, add blood feeding, pain, stress, and potential movement of bloodborne pathogens. The takeaway is that behavior predicts risk: contamination, irritation, blood feeding, or pathogen movement. Fear not, this is not a memorization table. The point is that non-biting nuisance flies can carry many zoonotic enteric parasite stages. Think about where flies land: feces, decaying material, wounds, secretion, feed, water, and surfaces. In cases, the major concern is mechanical carriage. Parasite eggs, oocysts, cysts, larvae, and other infectious stages are moved from contaminated material towards feed, water, animals, people, or fomites. The fly is not always part of the parasite's life cycle. It may simply be a tiny mobile contamination event with wings. But here's the nuance: carried by flies does not always prove the same transmission route. Some parasites can be transferred by eating the fly itself, which starts the overlap with transport host or paratenic host thinking. Other parasites require true intermediate hosts, where development occurs within the arthropod. So use this table as a risk pattern table, not a life cycle table. The clinical message is environmental. Fly control is not just about annoyance. It is also about sanitation, manure management, feed hygiene, and reducing opportunities for parasite stages and other pathogens to move through the environment. Now we move on to Glossina, the tsetse flies. This is one of the cleanest examples of a Cyclorrhaphan as a biologic vector. Tsetse flies transmit several species of Trypanosoma, and that parasite undergoes development within the fly before transmission. In humans, the classical disease is African sleeping sickness. In animals, the broad syndrome is called Nagana. The major veterinary trypanosomes include Trypanosoma congolense, T. vivax, T. brucei, and T. simii. The zoonotic species T. brucei gabiense and T. brucei rhodiense connect this topic to One Health. For this overview, do not get trapped into every species row. The main idea is the vector relationship. Glossina is not just moving contaminated blood mechanically, it is part of the parasite's life cycle. Now we move on to the family Hippoboscidae, the keds and louse flies. The etymology helps. Hippos points towards horse, and boscin means to feed or graze. These are tough, flattened, host-associated flies built to cling to animals. Clinically, the big pattern is close host association and blood feeding. These flies do not simply visit the host for a quick meal and leave. They are built to stay close, cling tightly, and exploit the host over time. A classic veterinary example is the sheep ked, Meliphagus ovinus. This species is wingless and spends its entire life on the host. That close association matters because keds can cause irritation, blood loss, wool or hide damage, and in some settings, they may also be associated with parasite transmission. So when you think Hippoboscidae, think flattened clinging flies, close host association, and blood feeding. And one more thing makes this family especially memorable. The reproduction is wonderfully strange. The reproduction of keds is one of the most memorable features of this group. Like Glossina, the tsetse flies, Hippoboscids use adenotrophic vivipary. Let's define that term. Adeno means gland, trophic means nourishment or feeding, and vivipary means bearing. So adenotrophic vivipary means that the female retains developing larvae inside her body and nourishes it with secretions from specialized milk glands. This is very different than the usual mental picture of a fly laying many eggs into the environment. Instead, The female carries a single developing larva internally, feeds it with milk gland secretions, and then deposits it as a mature larva. That larva quickly pupates. If we anthropomorphize just a little, this is part of what makes these flies feel so strange and memorable. The female is, in a loose sense, carrying her young internally and providing a milk-like nourishment before release. It is not mammalian pregnancy, of course, but it is an unusually intimate and highly specialized reproductive strategy for a fly. The key teaching pearl is that Glossina and Hippoboscidae share this same broad reproductive pattern. This table reviews several biting fly examples. Stable flies and tabanids can transfer blood-borne agents. Tsetse flies and some hippoboscids are biologic vectors because parasites develop within the fly. The table also reminds us that flies are rarely the only route. Ticks, iatrogenic transmission, other vectors, even terrifying vampire bats, and direct management practices may also matter. The clinical habit to build is this. When you see a biting fly, ask whether its role is contaminated mouthparts, repeated blood feeding, true biologic development, or some combination of those patterns. Now we switch from blood feeding to secretion feeding and non-biting vectors. This is where we can accidentally make the wrong shortcut. Non-biting does not mean irrelevant. Face flies are classically linked to Moraxella bovis. Some non-biting or secretion-feeding flies can also act as intermediate hosts for nematodes and cestodes. Houseflies can be involved in the life cycles of Habernema and Droscia. Fortica species are important in the Thalasia calipeda story. The takeaway is that secretion-feeding behavior can create real veterinary risk. Ask not only does the fly bite, also ask what fluid or surface is this fly seeking. Drosophilidae gives us the fruit fly exception. The name Drosophila means dew-loving, which is a strangely elegant name for flies we often associate with fermenting fruit and organic material. Most Drosophilads are not important biting flies. That is why this image matters. Most zoophilic Drosophilids feed on tears or ocular secretions, and that behavior can support transmission of eyeworms. Fortica species are an example because they can transmit Thalasia calipeda. Vector biology is not always blood biology. Sometimes the key ecological question is whether the fly seeks moisture and secretions around the eye. Now we move into myiasis. The word comes from mya, meaning fly. Thus, myiasis means infestation of a living vertebrate by fly larvae. That definition matters. The adult fly may be a visible nuisance, but the larval stage is usually the one causing tissue-level damage. We group clinically important Cyclorrhaphans by larval pattern that we use in practice. On the left are the maggot-associated families, Muscidae, Calliphoridae, and Sarcophagidae. Muscidae includes familiar nuisance and biting flies, although selected species have been associated with myiasis. Calliphoridae are the blowflies, and Sarcophagidae are the flesh flies. The name Calliphoridae means beauty-bearing, which fits the mesmerizing metallic blue and green bodies of many adult blowflies. But in veterinary medicine, these beautiful flies have ugly consequences. Their larvae may be involved in wound contamination, facultative myiasis, and in some species, serious obligate myiasis. The etymology of Sarcophagidae hits almost too perfect. Sarco means flesh, and phage means eating. That makes flesh flies a name everybody can remember. On the right is the bot pattern, represented here by Ostrinae within the family Estridae. Bots and maggots are both fly larvae, but in veterinary medicine, we often use the term bot for the more specialized larvae of estrid flies, such as nasal bots, warbles, and stomach bots. These terms help us organize different families, life cycles, host sites, and clinical consequences. Obligate versus facultative myiasis is about the biology of the larva, not just the family name. Estrid bots are classic obligate parasites, but important obligate myiasis producers also occur amongst blowflies and flesh flies. Other species are facultative and take advantage of wounds, necrotic tissue, or contaminated sites. In summary, Muscidae, Cyclorrhaphan, and Sarcophagidae often give us maggot-associated myiasis. Ostrinae gives us bot-associated myiasis. When looking at these flies, always ask, is the larva an obligate parasite, or is it taking advantage of damaged or diseased tissue? This is another dense reference chart, so let's read it as a pattern. Muscidae are usually not the classic obligate myiasis family, but some species can be associated with wounds. Cyclorrhaphan include important blowflies, including New World and Old World screwworms, which are key obligate traumatic myiasis names to remember. Sarcophagidae are flesh flies. Several are important in wound or cutaneous myiasis, and many are larviparous or oviviparous, meaning larvae may be deposited or hatched very quickly. That reproductive strategy can make tissue involvement feel sudden. Ostridae are different. These are botflies. The adults typically lack functional mouthparts and do not feed. Their larvae are obligate internal parasites. So remember, larvae cause myiasis, families differ in biology, and screwworms are high-stake recognition targets. In the laboratory or hospital, posterior spiracles can help narrow larval identification. Spiracles are breathing openings, and in many Cyclorrhaphan larvae, the posterior spiracles change as the larva grows. This is where the terms instar and ecdysis matter. An instar is a developmental stage between molts. Ecdysis is the shedding of the outer cuticle so the arthropod can move into the next stage. So when we say first instar, second instar, and third instar, we are describing larval stages separated by molts. As the larvae move through these instars, the posterior spiracles often become more developed. The general rule is that the slit number increases with the instar. First and second instars may have fewer or less developed slits, while third instars commonly have 3 slits. But as always, there are exceptions. Use spiracles as part of the pattern. Combine them with body spines, mouth hooks, larval size, site of infestation, host species, and geography. The goal is to know what features matter when someone hands you a maggot. New World screwworm, Cochleamia hominivorax. It's a high-stakes recognition topic. The species name is unpleasant, but memorable. Hominivorax points towards eating humans, and it reminds us that these larvae feed in living tissue. In veterinary medicine, the major concern is livestock, pets, wildlife, and occasionally people. This image shows several recognition clues. Creamy white larva with bands of dark body spines, dark oral hooks at the anterior end, and pigmented dorsal tracheal trunks visible posteriorly in mature larvae. Those features are very important when evaluating suspect myiasis cases. Recognize suspicious larvae and report promptly. Do not make the final diagnosis casually from a slide image or a glance at the wound. Suspect cases need proper submission and confirmation through appropriate animal health channels. Now we step into family Estridae, the botflies. The name estrus is wonderfully dramatic. It comes from Latin and Greek origins associated with a gadfly, a sting, and the frenzy or agitation that tormenting flies can create. That's a pretty good emotional description of how animals may react to botfly activity. Adults often have vestigial mouthparts, therefore they cannot eat. The larval stage is the clinically important stage. The family can be organized into 4 practical subfamilies. Cuterebrinae are the warbles or skin bots. The name Cuterebra means skin drill, and that fits the clinical image of a larva developing under the skin and creating a warble with a breathing pore. Hypodermatinae are also warbles, often called heel flies. Estrinae are nasal bots, and gastrophilinae are stomach bots, especially in horses. This symbol organizes the larval by system, such as the integument or respiratory system. Esterids are often host and site specific, and their larvae define the disease. The subfamily Cuterebrinae includes a couple prominent species. In North America, Cuterebra typically affects small rodents and lagomorphs, but aberrant infections can occur in dogs, cats, and humans. Flies will lay eggs at the entry of burrows. The larva may invade through the nose or other attachment sites and then develop into warbles. In cats, Cuterebra is more than a skin warble curiosity. The most common presentation is still a subcutaneous swelling with a breathing pore, but aberrant larval migration can cause serious disease. Migration through the central nervous system has been associated with feline ischemic encephalopathy and rare ocular migration that can cause severe inflammation, uveitis, blindness, and in some cases, the loss of an eye. That is the kind of detail that turns a botfly from trivia into clinical relevance. Another famous member of this subfamily is Dermatobia hominis, the human botfly of tropical regions of the Americas. Instead of laying eggs directly on the host, eggs are attached to a carrier insect, commonly a mosquito. When that carrier lands on a warm mammal, the eggs hatch and the larvae enter the skin. Fly cycles can be clever, host-specific, and visually distinctive. Let's look at the word hypodermatinae. Hypo means under, and derma means skin, so these are under the skin warble flies. In cattle, adult females attach eggs to hair, especially on the legs and lower body regions. After the eggs hatch, the first stage larvae, or L1, crawl to the base of the hair shaft and penetrate the skin, often through or near the follicular opening. From there, the larvae begin their internal migration before eventually becoming subcutaneous warbles. The path from skin entry to subcutaneous warble is not random. In cattle, Hypoderma lineatum larvae migrate to tissues near the esophagus, while Hypoderma bovis larvae migrate to the region of the vertebral canal and epidural fat before eventually moving back and forming subcutaneous warbles. This is the reason why knowing parasite patterns matters so much. Cattle are usually treated after the heel fly season ends, before larvae reach these sensitive internal sites, or before grubs appear on the back. If systemic treatment kills H. lineatum while it's near the esophageal tissues, severe inflammation can contribute to bloat. If it kills H. bovis while it is in or near the spinal cord, cattle may develop neurologic signs such as staggering or paralysis. With cattle grubs, treatment is not just about choosing the right product, it is also about choosing the right time. Now we end the family tour with Gastrophilus, the horse botflies. The name is useful. Gastro means stomach, and philos means loving. These are stomach-loving botflies of horses, although the exact larval site differs by species. Adults have vestigial mouthparts, so do not feed. Females attach egg to horsehairs almost like nits. Strictly speaking, nits are lice eggs, so the more accurate term here is botfly eggs. But the visual comparison is helpful. Small pale eggs attached to hairs, waiting for the horse's licking, grooming, or irritation response to help move the larva towards the mouth. The egg deposition site is a major identification clue. Gastrophilus intestinalis lay eggs on the forelegs, and the larvae are associated with the non-glandular stomach. Gastrophilus nasalis uses the intermandibular space with larvae in the duodenum, and Gastrophilus hemorrhoidalis is associated with the lips and later the rectum. Remember the durable pattern. Egg site on the horse plus the L3 site in the horse gives you the life cycle clue. In summary, first recognize the group. Cyclorrhaphans are higher flies with short antennae, often an arista, and adult emergence through a circular seam. Second, ask how the fly behaves. Can it spread disease or cause direct damage? Third, predict the clinical problem. Know your taxonomy. Muscidae means nuisance, biting, and contamination. Glossina and Hypoboscidae bring biologic transmission patterns. Californidae, Sarcophagidae, and Estridae bring myiasis and botfly disease. And Drosophilidae remind us that tear feeding can matter too. If you identify the fly, understand the behavior, and connect it to the host, you can often predict veterinary problems. This can maximize your prevention and treatment strategies. Thanks for watching. I'm Christopher Lee, and what you know today protects everyone tomorrow.